Today I'm going to present you a couple of things that I've been doing the last few years. I've been using a lot in biological systems, solution state NMR and solid state NMR. Why solution state? Because most of the time we look for soluble system that can be closer to cellular environment. Usually we can analyze low molecular weight, such as metabolomics, and we can apply some specific instrumental labeling for high molecular weight and understand how the protein machinery can work. For the solid state NMR side, I'm going to present some data about high molecular weight system, typically insoluble or very poorly soluble, and the application can be run on microcrystals of these proteins and sedimentation. So let's start with the first part of the talk. I've been working while I was in UK on antibiotic resistance. Since, as you can see, there is a high demand for new efficient drugs, and the forecast that by 2050, 10 million of deaths per year can arise from antibiotic resistance mechanism. There are some critical priority to highlight here. One is the Acinetobacter baumannii that is already resistant to carbapenem antibiotics. There is Pseudomonas aeruginosa and Enterobacteriaceae bacteria. So in this case, these are key elements to keep an eye for. The antibiotic discovery timeline, as you can see, started late 20s of the previous century, but basically since 1993, it's slowed down a lot. So a lot of new class of antibiotics is needed at this point. And as you can see, I will focus most of my talk on Acinetobacter baumannii. It's typical strain that is found in healthcare institutions such hospitals. And a lot of countries are already reporting a large number of this strain resistant to carbapenem, okay? What we can do as a scientist, we can look for new synthetic pathways, or mostly and more efficiently look at class of antibiotics produced by bacteria. For example, erythromycin or vancomycin. These are the most currently used antibiotics and are coming from natural products or their derivatives. So nature, in this case, provides a great inspiration for this case. How nature is organized and works for this system is exploiting two different systems, NRPSs and PKSs. NRPS stands for non-ribosomal peptide synthesis, and PKS stands for polyketide synthesis. Typically, this huge machinery, because are really huge, megadalton assembly, are modular and amenable to engineering. As you can see here, how this particularly NRPSs and PKSs system are set up. There is a loading. There is module 1, module 2, up to module 10. Each module can do a specific function. So if we able to engineer part of this system, we can produce new classes of antibiotics or new classes of natural compounds. So typically have collinear genetic organization, highly modular, as I already mentioned. The main objective is, can we engineer the system and yields improved and protein-protein interaction can be understood completely? The short answer is yes, but I'm going to show you throughout the presentation today. So how antibiotics are produced is produced in NRPSs. NRPSs work basically as assembly line. Each station performs a specific function. Here I report, for example, cyclization domain, condensation domain, and adenylation domain. And there is this particular peptidyl carrier protein it's the connection between two different enzymatic reactions. Typically, this carrier protein, it's a small 4-helix bundle. There is a serine to which is attached to this phosphopanthetin. It's basically the branch of the arm that is transferring the substrate between the units. So let's have a look how this unit works. For example, we have the adenylation site where we have isoleucine, cysteine, and leucine. And then we have the adenylation domain is transferring the isoleucine to the carrier protein. Then there is a cyclization, and then there is a transfer, condensation. So in this case, you can see how the system assemble a new compound. In this case, the bacitracin from Bacillus subtilis. It's a new class of compound, okay? But this idea can be exploited also for antibiotics. Absolutely, yes. This antibiotic, N-acetylocin 2A, is the only arm we have against the resistant Acinetobacter baumannii acquired in hospitalization. So as you can see, these antibiotics can inhibit the bacterial protein synthesis and is active with the most common multi-drug resistant hospital acquired infection of Acinetobacter baumannii. So where does this antibiotic works? On the elongation factor, TU, and the acetylocin complex inhibit this complex. How is assembled? As you can see, there is a load module, module 1, and so on till module 10. So where we have the acid carrier protein, let me put a pointer so I can point here. This is the region I'm going to focus on because this can be engineered. Why we need to engineer this? I have to mention that this is an ester. This ester, it's very labile. So if we manage to use a new system and manage to change this ester to an amide, we made a bit more resistant. So we want to engineer the system to switch to this oxygen to a nitrogen, okay? So as I mentioned, we're gonna look into more N-acetylocin trans AT, PKS, and RPS hybrid module, where there is the 5917 PCP. From now on, I will call the 5917 PCP protein, and the C15, because it's 5915, is the condensation domain. In this case, the acceptor molecule, it's the variation of shikimate. The donor molecule is coming from all the 10 modules before, and this can be the final step of the producing an acetylocin 2. Okay. So let's have a look immediately. We did the solution state NMR structure characterization of the PCP17, where we managed to see where the serine sits and the phosphopanthetin arm is attached. As you can see, the classical 4-helix bundle is still there. The difference between other systems, we found that there is the disorder N and C termini. How we managed to get this information? Through 15N relaxation, where typically the 15N R1, R2, and heteroleucrar NOE are immediately telling us that they are disordered. So you can see the dip of the graph in this case and the height of the graph in the R1 case. You can see also the N terminal, it's higher, lower, and lower. So basically, we can focus on this region mostly, where the rigid part is, okay? At this point, we have the structure. We know that it's flexible, but we saw that we need the NMR titration for us. So we were able to titrate the PCP17 with a C15 protein and see which residue are mostly affected by the interaction. As you can see, globally, the entire protein is a bit affected, but at the same time, the highest region where there is a change is the C-terminal. I reported here in red the residue that are interacting with the protein. So you can see that there is the phosphopanthetin arm, and there is a lot of residue in the so-called hydrophobic core. This is not so strange in this case, but we will see later. We were able also to see, using the chemical exchange saturation transfer, which residue and which conformation are available in this case. As you can see, we were able also to evaluate and measure the dissociation constant. That was 0.7 micromolar. So I would say it's a weak interaction involving the globular domain, but it's very tight in terms of the C-terminal. Just to show you how this process is taken care of, usually we measure the PCP alone. We add the C15. In this case, just to prove that there is interaction only with the C-terminal domain of the PCP, we also run the BHD domain on the C15. You will see later on actually this beta-hairpin docking domain, because it's a key mechanism of interaction between these two proteins. So you can see the C-terminal region is the one strongly interacting. So the chemical shift was very large in this case. Okay, so this is for the solution part, but the complex, I forgot to mention, it's the protein by itself, the PCP17, it's 10 kilodalton, a bit less actually, but it's, let's call it 10 kilodalton. But the big protein is 55 kilodalton, the C15. So in this case, we reach a complex of the size of 70, about 70 kilodalton, okay? That makes almost impossible to look into solution the C15 side. So this is why, while I was in UK, we approached the the solid state NMR, mostly carbon and nitrogen and proton acquisition. Why solid state though? Basically, we can explode different spinning speed. This means different approaches. So usually for very large system and large sample available as slow spinning, usually we're gonna use carbon detection. In case of dilute option for protons, in case of per-detected sample, we can still use this range of speed. But most of our sample has been running on from 67 up to 111 kilohertz. So this is 111,000 rates per minute, okay? Per second, sorry, okay? Next, we perform this solid state NMR sedimentation of this complex. The complex, as you can see here, it's 70 kilodalton. We back exchange all the protons that are exchangeable in the protein, so mostly the amides of the PCP17 with not labeled C15. We span our protein to 60 kilohertz at a magnetic field of 700 megahertz. I have to mention that it's highly dynamic complex, and in spite of extensive trials, does not crystallize. We tried extensively, but we couldn't crystallize. So you can see the quality of the spectra. So solution state NMR, nice and sharp peaks. The CP-based solid state NMR, a bit overcrowded and not so nice. The inept-based experiment, so basically the solution state NMR experiment apply in the solid state, show basically few signals. The main question we had at this point was, can we get more information spinning faster? Obviously, the answer was yes. The quality of the spectra improved. It's not yet the most beautiful one. You can see the correlation 1H15N. You can start to see the splittings. You can see the interaction. Keep in mind that this is the solution plus BHDD. The CP-based, a solution with no BHDD. So you can see already the sum of the chemical shift, in particular for the valine 284 and the arginine 315, matching the one that we have in solution. But to be sure, absolutely, we need to assign the entire protein. So we run all the CP-based correlation for carbon. So basically, this is the solid state version of the HSQC. Then we run the solid state version of the HSQC, exploiting the inept transfer. And still, you can see how much is improving the quality of our spectra. One thing that should come immediately to your mind that the C-terminal is bound in the same conformation as for isolated BHDD. So basically, this is a strong confirmation that the C-terminal is the main actor in this case, and the BHDD domain is the main actor for the C15 side. We pushed the limit and we went for the TUCC experiment, where we can see all the correlation inside the protein through bonds. Some assignments were easily transferred from solution state. For some, at least we can tell which is the minimum change in chemical shift. So being able to work with 13C fully protonated system, it's a game changer in this case. And the assignment is still in progress, takes a long time because it's not the easiest way to assign this protein. But so then the question is how we can map this interaction on C15 and PCP17 without excluding the conformational change. We need to understand which residue are changing, okay? And this confirmation in solid came immediately up with a C-terminal, like in solution, and a lot of residue in the hydrophobic core. With only 2 days, we cannot really progress. And to look for these changes, we decide to go for the easiest way, molecular dynamics. So let's try to see which data can correlate in molecular dynamics. In this case, we did the analysis of the NMR titration residue and the C-alpha chemical shift changes. The animation, keep in mind that it's looking on the first mode of the principal component analysis. You can see here how the motions are start to correlate each other. So the C-terminal and the hydrophobic left, it's twisting. So the conformational rearrangement in this case involved the rotation and translation of helix 1 and loop 1. If you look where the setting is, it's up here. So this is consistent to a conformational rearrangement influenced and mediated by the C15. So to conclude this part of the talk, we discovered that the C-terminus is responsible for the interaction. The intermolecular interaction involves disordered part of the protein and a smaller part in the first step of the C15. And we need to combine several techniques because there is a bit of X-ray in terms of C15. There was mass spectrometry, molecular dynamics, molecular biology. There was also this technique applied for mass spectrometry, the carbon footprinting, and solution state NMR. So here I wanted to show how the NMR alone cannot do the entire work, but needs to be integrated in a broader selection of technique. From this side, I want to thank all the people from UK, where I've been staying for 5 years. And now let's switch a bit to the latest work I've been doing using NMR. metabolomics. So I've been involved recently in characterization of human cytomegalovirus for infants, and we discovered that this is ubiquitous. IgG seropositivity, it's between 60 and 90% of adult. As you can see, it involves mostly the western part of the world and the eastern, with some spots in Africa, and is correlated sometimes with socioeconomic condition. There is high prevalence in South America also. What is this virus doing? To immunocompetent individuals, most likely it's asymptomatic, or mononucleosis-like syndrome. In case of immunocompromised individual, systemic disease, serious complications sometimes are fatal. Brain, such as encephalitis, retinitis, lung pneumonia, and so on. But we focus most of our work on congenital infection. This congenital infection, it's a huge problem because it has been reported that 10% of babies are symptomatic at birth, while 90% completely asymptomatic. 75% have no complication. 15% might develop long-term complication in case of asymptomatic babies at birth. If we look at the situation of symptomatic babies at birth, there is a huge, high 10% mortality rate, 60% survival with severe to long complication, and 30% only without any complication at all. This virus, it's multifaceted, as you can see. It's an enigmatic virus for many reasons. In particular, the clinical outcome of congenital infection can range, as I mentioned, from asymptomatic to severe disease. And this variation depends on a combination of factors, including host genetic background, viral genetic variability, phenotype, and the host antiviral response, okay? These severe complications that usually are led by this virus are hearing loss, microcephaly, hepatosplenomegaly, petechia, visual impairment, development delays, motor delays. The severity of the symptom is strongly associated with timing of maternal infection during pregnancy, with the most severe clinical outcome if the infection occurs during the first trimester of gestation. So we designed the study considering this scenario. We have enrolled a cohort of newborns with congenital cytomegalovirus infection and conducted a series of catheterization involving various specialists, from the viral pathogenist lab in Torino, from some people from Sapienza in Rome, and the University of Torino Chemistry Department has taken care of metabolomics analysis. Okay. So we know already where some of the pathway in metabolomics can affect our virus. So you can see here where most of the virus might act. So we immediately dig into NMR experiment, and this is an example, the 1H NMR spectra we collect. The red are the patient, the controls, and the black are the controls. Sorry, the other way around. This typically is the aromatic region and the amide region. Typically, this is the amine, alcohol, and aliphatic regions, and usually this is the methyl region. Okay. We were able to identify 55 metabolites. Most of them were used in the analysis. Obviously, we reference our experiment to the TSP, and we apply the typical protocol for urine sample. And immediately we performed the PCA analysis. We were a bit surprised in the PCA's analysis because there was some kind of separation already. We have a larger number of controlled patients and a lower number of controls. Actually, we're pushing these limits a bit for the analysis, but we need more controls. The 1H NMR spectral dataset were first mean-centered, and then the analysis was performed. We cover only 56% of the total variance, but it's promising. We decided to go for a more detailed analysis using the partial least square discriminant analysis, PLSDA, and we were able, using two latent variables, to correctly assign 32 patients out of 35. Instead, using 3 latent variables, we were able to assign 34 out of 35. These results, obviously, it's a bit impressive, strange to this case, but I have to mention that most of these infants were collected between the 2-3 weeks after the birth. So the feeding, it's much more controlled, is less variable than adults. So in this case, was a bit easier. From this metabolomics profile, we were able to identify few metabolites that are typical of control in this case and typical for patients. And we were able to clearly identify betaine, formate, glycolate, lactate, succinate, threonine, citrate, acetoacetate for the patients. And this is typical in this case, of virus at least. And we were able also to see some of residue of metabolites typical for controls. So in conclusion, from this side, even though we were aware that a larger patient cohort is needed, and we need to further validate this study, indicates a genetic variability and in vitro phenotype might play a key role in influencing the clinical outcome of the congenital infection. Additionally, and I want to emphasize that there is a metabolomic fingerprinting on the congenital infection, and the study at this stage is going to get expanded soon into a multi-center where the University of Varese, University of Naples and Rome will contribute with more sample. And with this, I would like to thank the people in Torino, the collaboration we have with the Pediatric Sciences and with the Polytechnic University of Torino, and you for your kind attention and patience. So I'm happy to take any question.